Excellent. Thank you so much. This has been a really insightful on our section on HSV. I want to move on now to lipid nanoparticles. And Karen, so there has been a lot of momentum around LNPs lately, not just for RNA delivery, but expanding into broader applications across gene therapy. And so I'd love to get your take on where LNPs are providing the most value today, especially compared to other viral vectors. So or excuse me, to viral vectors. What are the most critical advantages of LNPs over viral vectors for gene delivery, particularly in terms of transient expression, re-dosability, and immune profile?
Yeah, so as you said in your question, they're incredibly useful for when you need transient expression of something. So, for example, if you're expressing genome editing components and looking to do that, that's one area in which I think it's been incredibly popular lately. In addition to that, though, because they seem to be non-immunogenic, it allows you to potentially to titrate in your dose. So for certain viral vectors, especially AAVs, for example, if you're doing a gene therapy with them, you build up such a strong neutralizing antibody response, typically you can't redeliver it in the same way to titrate up. So you're kind of one and done. Here you can potentially dose. And if it doesn't have a large enough effect, you can go back into the same patient and redose. And so that is the potential for a huge advantage. In general, these are thought to be non-immunogenic. Again, it depends on exactly how they're constructed. Are you adding targeting moieties that could be immunogenic? There's all sorts of reasons why you could potentially have you could generate that. But for the most part, I find them very useful for these sorts of areas where you want to redose or you want to express for a certain time frame, may not want that for a while, and may want to turn it on again.
Great, thank you. How can one tackle the formulation challenges of LNPs to improve targeting specificity and reduce off-target accumulation in non-hepatic tissues?
Yeah, so for some of these LNPs, right, you're trying to reduce accumulation non-hepatic tissues. For others, where you're trying to target non-hepatic tissues, you don't really want it to accumulate in the liver. So what people are doing are adding targeting moieties, which does make manufacturing a little more complex, of course, but can be done. But also simply altering the lipid composition, the surface characteristics of an LNP can alter its targeting. So there's a lot of work that's been done there, being done there in the process of it. And in addition, one of the interesting things is that they're not all very efficient at escaping the lysosome. So once it's internalized, you need lysosomal escape in order to release your payload into the cytoplasm. So there are some improvements on that can also help reduce the doses needed, which then reduce the amount of vector that's going to go to off-target tissues.
Good, okay. And speaking of payload, are there, and I know we talked about at the top of this section that, they're not just for RNA delivery, but expanding. And so are there emerging use cases for LNPs in delivering DNA, not just RNA? And what does that change in development approach or payload design?
Yeah, so because DNA is inherently more stable than RNA, it can be easier perhaps to get stability of your final product. And so that's one advantage it can potentially give you. Well, also what it will give you is potentially somewhat longer gene expression than a single dose will give you with mRNA, because the mRNA, of course, degrades more quickly once it's released into the cell. The DNA might be there for a little bit longer, and because it's actively expressing RNA, you then get, protein expression from there. So if it's something, a disease where you want to get somewhat longer gene expression, it can be useful to deliver DNA rather than RNA.
Okay, thank you. As always, I want to make sure we talk about manufacturing. Can you share some insights of how CMC and regulatory requirements for LNP-based therapies differ from those of viral vectors, and particularly with perhaps respect to analytics, stability?
Yeah, so the analytics are somewhat different, which is very interesting. And so one of the things that's been emerging in the field is that there's a need for more development of in vitro assays to really assess LNPs. And again, this will come with time. I'm sure that will come in there, but the in vitro platforms will look for efficacy and safety. So pretty much with the LNPs, you're looking at your consistency of the product. So, are they the same size? Do they contain the same components? How efficiently has your payload been encapsulated? So, it's particle characterization, encapsulation efficiency, and impurity profiling. And so those impurities will be different with an LNP that's been manufactured in the absence of cells, whereas viral vectors are typically manufactured in cells, in mammalian cells. And so there are other different types of impurities that you're looking to test. So in stability, looking for degradation products of both the LNP and its cargo. With viral vectors, you're really looking at stability, its long-term viability and function of that viral vector. So there are definitely some differences in the total in the types of assays that need to be done in order to characterize them fully.
Great. Okay. Before we move on to our audience questions, I do have one more question actually for both of you. And Karen, if you could please pose it to LNPs, and Francesca perhaps to HSV adeno. From your perspective, what are the most significant regulatory validation hurdles that remain for the various delivery systems? I know that's a big, giant question, but at the same time, if you could just give us a little bit of give us some tidbits.
Sure. I mean, I think there's the redosing question with LNPs, which I think they are, most will be redosable, but that needs to be shown clinically. They also need to be more scalable to get to commercial. So again, with every new development, you need to be sure to be able to scale them. And also checking, again, developing their efficiency. So whereas when we use viral vectors, right, they've evolved naturally to deliver genetic material to cells, LNP is an in vitro creation. So again, making sure it's sufficient enough for the doses you need to give will be one of the areas that will be interesting to watch evolve.
Sure, thank you. And Francesca, yeah, same question for you from a regulatory perspective.
So I think from the regulatory perspective, the FDA is getting more and more used to see the use of these vectors, especially in the oncology space. I think for us, it would be the possibility to have a platform approach, for example, to our HSVs, because if you make the vector is the backbone of the vector is what drives the biodistribution of that vector. And our approach, especially with our new platform Enlighten, is to change the genes that you can, that you can express. And so the possibility of not having to repropose every time a full tox and biodistribution package, considering that you have probably a fundamental approach because the vector is the same in terms of biodistribution. I think that will be extremely great, and that will facilitate not just our approach but also others. The other hurdle is the CMC, right? So we all know that in these biologics, the process is the product. And so I think that speeding up the review and understanding certain changes that are natural to the history of the development of this in terms of process development, I think will be great. But I think the regulators are trying to understand, and I think there is a drive towards getting medicine to people faster, especially medicine that have proved clinical activities in clinical trials.